Academy of Science. He has received DST SERC Fast Track Young Scientist Award, and his main research area is maintenance of genome stability in plants and molecular mechanism of DNA damage repair in the context of environmental and genomic traces. He is going to talk about Arabidopsis rad21.3, yeast ortholog of sister chromatid cohesion 1 protein repairs, which repairs double-stranded DNA breaks via homologous recombination mediated pathway and remodels damaged chromatin. Dr. Roy, please.
Thank you for the kind words of introduction, and good afternoon to everyone. So first of all, thanks to the organizer for giving me this chance to present my work in this platform. So I'm going to talk about the role of a RAD group of protein. This is specifically RAD50 or 21.3. So for understanding the mechanism of homologous recombination mediated DNA repair, particularly the double-strand breaks in plants, to further understand the homologous recombinated mediated mechanism in a better way in the plant genome. This one. So plants are actually widely exposed to various environmental and abiotic stress factors, and some of which are often responsible for induction of the different types of DNA lesions, and particularly double-strand breaks, which often responsible for the induction of genome stability and therefore compromises plant growth and development. So under this condition, plants have developed an extensive array of mechanisms to actually get rid of all these things, and this is involving the activation of very extensive and robust DNA damage response pathway. So that is associated for the rapid detection and the repair of double-strand breaks, and also different types of breaks in the plant genome for maintaining of the genome stability in plant system. So for maintaining the genome stability, so this is also very actively involved and very intrinsically associated with maintaining this, the stability of the chromosome, the stability of the chromatin structure and the chromatid structure, because the repairing of the breaks also that involves remodeling of the damaged chromatin and for the proper alignment and for the proper segregation of the chromatids. So just to avoid the segregation-related abnormalities for the faithful segregation of the genetic information. So within this context, cohesin, so this is one of the very important part of the interchromosomal structure that is responsible for holding together the sister chromatids and for the faithful segregation of the chromatids during the separation. So the cohesin is a large multi-protein complex and contains, apart from the structural maintenance of chromosome, that is SMC1 and SMC3, that contains two alpha-glycine components. So one is the sister chromatid cohesin 1 and another is sister chromatid cohesin 3 protein. So I'll be talking mainly about the SSC1, or this is sister chromatid cohesin 1 is also known as RAD21 protein. So Arabidopsis genome that contains 3 homologs of the RAD21.3 proteins, or the genes better to say, 21.1, 1.2 and 1.3. So most of the cases it has been found that the protein, they actually differ from their amino acid composition, mostly at the level of the N-terminal amino acid sequences, but previous studies have indicated possible function of this protein in mediating DNA repair via homologous recombination pathway. And most of the cases they have given emphasis on the role of RAD21.3 and 21.2, and also in many different plant developmental processes. But specifically, still we do not know about the functional involvement of RAD21.1 and 3 in repairing DNA breaks, maintaining chromatin structure stability, and regulating cell division. So for this, for understanding the biological function of this RAD21 groups of genes, so first of all, we characterize the mutant lines of this RAD21.1, 21.2, and 21.3. And we specifically, from the initial stages, we found there is a retarded growth response of 21.3 mutant lines. And when we initially screened for the genotoxicity and abiotic stress related agents by using all the 3 mutant lines, we found that RAT 21.3 specifically showed enhanced response in comparison to 21.2 and 21.3 two mutant lines. So this is the responses of 21.3 mutant lines is in comparison to 21.1 and 21.2 mutant lines. And we then specifically found that, so we then identified the genetic interaction between these three alleles. So RAT 21.3 mutant line exhibit increased sensitivity when we challenged with the dehydration stress, that means with the PEG treatment, and that leads to the water deficit challenge. So under the germination level and also the primary root growth response were compromised in RAT 21.1 and RAT 21.3 mutant lines in comparison to 21.2 mutant lines. So then we for the genetic interaction studies, we developed the double mutant lines of RAT 21.3 with the 21.1 and 21.2, considering the increased sensitivity of 21.3. So we found that 21.1 and 21.3, they show further hypersensitivity in presence of the PEG treatment in comparison to the 21.3 and 21.2 double mutant line. And basically the phenotype of 21.2 and 21.3 mutant line was closely related to the 21.3 single mutant line. So based on that, we initially concluded that RAT 21.3 and 21.21 that actually function synergistically under the dehydration stress. On the other hand, RAT 21.3 is actually functioning epistatic to RAT 21.2. So then we specifically given emphasis to understand the function of RAT 21.3 in Arabidopsis under the dehydration challenge. So for this, we actually developed different Arabidopsis lines with the genetically different Arabidopsis line that with the altered expression levels of this RAT 21.3 with overexpression line, complementation line, etc. And we found that the promoter activity of this gene was found to be significant in case of both mitotic tissues and the meiotic tissues. And when we specifically identified the phenotypes of these mutant lines, under the control condition, it shows different types of growth defects. For example, the first and the second leaf of the mutant are shorter than the wild type lines and the overexpression lines, and 8 and the 9 leaf of the rosette were abnormally larger in size. On the other hand, the total number of rosette leaves that remains most of the cases unchanged in the mutant lines. Now, apart from these growth defects, we also found, so there are growth defects in the rosette leaf diameter and compromised growth responses of the mutant line in comparison to the overexpression line, which somehow compensated the compromised growth responses of this mutant line. But apart from this, the mutant line shows disruption of the cruciferous floral organization of this as compared to the wild type plant. And apart from this, we found developmental defects in calyx and also in the ovule formation. So altogether, so this indicated that loss of function of RAT 21.3 that leads to different types of developmental defects, and therefore indicating its possible function in regulating various developmental responses. Importantly, so we also found that there is a kind of a delay in flowering response in this mutant line. So this is kind of quite similar as in case of the Brahma and other well-known chromatin remodeler mutants. So specifically, when we studied the responses of this RAT 21.3 mutant lines in presence of the challenges, increasing level of challenges in the PEG treatment or the extensive level of the water deficit condition. So these mutant lines show the compromised growth response during the germination and the post-germination seedling establishment. Apart from this, we found that in the wild type plant, so the RAT 21-23 protein accumulation was significantly increased under the dehydration challenge, and the wild type plant shows significant level of increased RAT 21.3 foci formation under the dehydration stress. But when we use the RAT 21.1 and 21.2 mutant background, so the foci formation of RAT 21.3 does not actually decline significantly. So from this, we conclude that RAT 21.3 probably functioning upstream of RAT 21.1 and RAT 21.2. So since the dehydration stress or the water deficit condition that actually operates through the induction of the oxidative response, and also that leads to the shoot apical meristematic cell death, we found that in the mutant line there is extensive level of accumulation of the ROS as compared to the overexpression line, and also the mutant line shows a reduced level of the root apical meristem cell viability under the low water potential stress conditions. So considering the compromised root growth of the mutant lines, and since RAD21.3 is a component of the cohesin complex, then we found that the mutant lines under the dehydration challenge that shows different types of chromosomal abnormalities, and those are specifically related to the chromosomal abnormalities under the separation conditions and the chromatid alignment. So from this initial data, so we concluded that RAD21.3 function is important for chromatin alignment and chromatid segregation. Then this was also associated with the enhanced accumulation level of double-strand breaks in the RAD21.3 mutant line, also slower rate of recovery, and this was also consistently associated with the accumulation of gamma H2X signal, so that is acting as one of the conserved signal for the accumulation and existence of double-strand breaks in the genome. So we found that, then we also used the double mutant lines, and we found that in RAD21.1 and RAD21.3 double mutant, so the phenotype was again drastically compromised, and there is also slower growth, root growth recovery, and they also slowly recovered the double strand breaks. So from this we concluded that RAD21.3 and 21.1 gene functions are equally responsible for repairing breaks in the DNA under the dehydration stress. And RAD21.1 and RAD21.3 double mutant, they also exhibit increased accumulation level of this gamma H2X foci formation. Both of this is when they were challenged under the under the dehydration stress conditions. But under these conditions, so the foci formation of RAD21.3 does not actually decrease. So this indicates that RAD21.1 and RAD21.3, they act synergistically in repair of double strand breaks. Now, considering the compromised cell growth and developmental responses in RAD21.3 mutant and other two mutant lines of RAD21 gene family, so small gene family with the three alleles or the three genes members, so they RAD21.3 mutation line that shows increased when they were challenged with the dehydration stress, though, so they showed increased nuclear population under the G0 subphase. And this was also consistent with other single mutant lines and also double mutant lines. So these lines, these results together indicates that the homologs of RAD21.3 acts redundantly in the regulation of the progression of the cell cycle, and when there is a loss of function mutation, so that leads to cell cycle arrest at the G2 to M phase of the cell cycle, and that could explain the compromised root growth of most of the mutant lines. So now we know that the double strand breaks are specifically repaired by the two major mechanisms. One is the homologous recombination and the NATJ mechanism. The homologous recombination mechanism is the most precise mechanism that leads to the restoration of the original DNA sequences. And we know that the RAD50 epistatic or RAD epistatic groups of protein, they specifically participate in repairing the double strand breaks by homologous recombination pathway. And this is very important pathway for the restoration of the original sequences and for the development of the new allelic combinations. So, and within this context, RAD54 is one of the very prominent member that is participating in repair of double strand breaks by HR pathway, as well as this is also acting as a very well-known chromatin remodeler to remodel the damaged chromatin at the site of damage. We found that RAD21.3 specifically interacts with RAD54 in yeast and also in the plant cell. And further molecular genetic analysis and immunological studies indicated that RAD21.3 co-localizes with RAD54 at the site of DNA damage under the dehydration stress. On the other hand, when there is a loss of function of RAD21.3, so that leads to decreased localization of RAD54 foci formation. So from this study, we conclude that RAD54 play an important role together with RAD54 in repairing double strand breaks. On the other hand, so RAD21.3 specifically function upstream of RAD54 and probably recruits RAD54 at the site of DNA damage. Now, just to validate this result, and since there are 21.3 mutant line that showed enhanced level of DNA damage, or better to say the double strand breaks and slower rate of recovery, then we asked whether this is, and also it interacting with the RAD54, one of the well-known player of the HR-mediated repair of double strand breaks. So we then asked that whether RAT21.3 participates in the HR-mediated pathway. So for this, we actually use the IC9C reporter line. So this line that contains the disrupted gas cassette, and when there is a homologous recombination event that leads to the reconstruction of the gas cassette, and this can be detected in terms of the gas activity and gas staining. So when you cross this RAT21.3 mutant lines with this reporter lines under the PEG treatment or the dehydration stress challenge, we found there's a significant decline in the homologous recombination frequency within this line. So these results indicated that RAT21.3 repairs double-strand breaks via the HR-mediated pathway. Now, since this is the component of the cohesin complex, and there is a possibility of role of this protein or the gene in participating in the chromatin remodeling, since it is also interacting with RAT54, so then we use RAT21.3 and RAT21.1 mutant lines in a series of fish assays, and we found that these two mutant lines shows enhanced level of sister chromatid separation during the interphase nuclei before the cell division. So this indicated that both the RAT21.3 and RAT21.1 act synergistically to maintain the sister chromatid cohesion complex and the chromatin structure stability. So subsequently, we found that RAT21.3 mutant line shows reduced bivalent formation in the PMCs in the metaphage cells, and also these mutant lines, the same mutant line shows that exhibit non-specific association with the centromeres. So these were associated with the after recombination event. So these results indicate that RAT21.3 function is important for remodeling of the chromatin after the recombination. So both RAT21.3 and RAT21.1 mutants, they show the delayed release of the cohesin. So this is in contrast what we found in case of the mitotic cells that leads to the early separation of the chromatids, but in the meiotic cells, so they actually showed the delayed release of the cohesin, and therefore that leads to several abnormalities during the meiotic cells. So these results indicated that RAT21.3 and 21.1 mutant also play important role in chromatid alignment and segregation for maintaining the chromatin structure stability. So the overall story so far that tells us that RAT21.3 loss of function that leads to various growth defects, and therefore indicating its function in regulating different growth and developmental responses. It functions synergistically to RAT21.1, but it is epistatic to 21.2. Both of 21.1 and 21.3 function in repairing double-strand breaks. Genetically, RAT21.3 function upstream of RAT54, but it recruits RAT54 to the site of DNA damage, and RAT21.3 repairs double-strand breaks via the homologous recombination pathway and remodel the damaged chromatin synergistically by acting with RAT21.1. So with this, I just conclude, and I would like to acknowledge, so my funding agencies, the mentor work has been done by Shomrat, so he's also over here for poster presentation. So my lab members and also my collaborators, Shaurabh, Professor Pukta, and Ibrit, and thank you so much for your kind attention for the talk.
The lecture is now open to any question from the students. None?
Thank you, Dr. Sujit. Very wonderful talk.
Thank you.
Do you actually have any data to support that chromatin remodeling is actually going on?
Yes, we have some additional data. So other
Or did you make a mutant with SWI/SNF, for example? Did you make a double mutant?
we have a double mutant with probably SW1 Swiss one, or so we also have the data that how it is remodeling the chromosome, that we have the several series of data with the micrococcal nuclease assay to see that. So it is actually responsible for chromatin remodeling by spacing apart the chromatins, the nucleosomes basically. That's what the mechanism sort of.
Great, very good. So any insight into why homologous recombination is so poor in plants? It's very, very, very low frequency.
That's what, because most of the cases plant cell, also in the animal cells, the mammalian cell, the somatic cells, they repair the double-strand breaks via the non-homologous end-joining pathway. So that is very error-prone. But most of the cases when it is comes question to the maintaining the stability of the genome, so because of the meiotic cells, so after the recombination, so during the recombination breakage and reunion, so early theory, so there they employ the HR pathway. And we know that most of the allelic frequency and diversity of the occurrence of the new alleles or whatever the genome editing programs nowadays we're having. So these are mostly relying on the exact mechanism of the HR-mediated pathway to actually accurately repair and replace or edit the genome. So that's what we are interested to know more about, the mechanism of the HR-mediated pathway in plant cell. Although the frequency of occurrence is very less, that we still do not know how it is. Yeah.
The same goes for example, in mice, it was very difficult to do.
Right.
But once the papers were published that you can have 5 to 10 kb of sequences, it was basically length. As you kept on increasing the length, you could achieve homologous recombination even in mouse somatic tissues. But in plants, doesn't matter what you do, you even put 5 kb or 10 kb, it's abysmal.
Right.
Almost impossible.
Right.
So something more is happening. HR is hugely suppressed.
Exactly. Yeah, that's also an important topic of research, that under which condition plant takes the decision that which pathway has to be employed for either HR or HEJ. That's also one of the important switch and the topic of active research under this field. Right. Yeah.
DDR response you see in the mutants only after challenging with some stress, not in the normal condition, right?
No, although in the normal condition, so normal condition, they still shows the compromised growth, and particularly the 21.3 as compared to the other two mutants. So, but when we challenge this with the PEG treatment or the dehydration, so the response or different other treatments, so NaCl or the cisplatin, the genotoxic.
Yeah, in response
agents.
to the stress challenges, of course, yeah. But
Yeah,
in the
so
normal condition also you see DDR response already triggered.
Yes, at the basic level. So they are showing the DDR response.
Okay, and one technical question. So those cell cycle specific stages, G2M and all, did you measure with the facts? So,
Yes.
and those were root
Yeah,
samples?
yeah.
So how you melt the cell suspension or what?
Yes. No. So I think with the root samples are processed and macerated, and probably the nuclei were isolated, and then we did that from the nuclei. Yeah. From the root cell nuclei.
So you are doing it in your lab?
Right. That is standardized already. Yeah.
So I have a similar question, and we had a lot of problem in doing this cell cycle stages,
Right.
and we could not get the right. But my question is this: the RAD 21.3 mediated DDR is specific to only PEG dehydration stress, or it can be in any stresses?
It could be any stresses, but here we see that RAD 21.3 or the other members that specifically showed hypersensitivity under the dehydration stress. That's why we then specifically expanded and extended the work by following up that stress condition.
So it may happen that under some different stress, other RAD may take over.
Could be, yeah, because under the field condition there are different stress conditions. There should be, right? Yeah. Thank you.
Are there any other questions? No, I have just a query. Did you ever try any ionizing radiation? Because that's one of the most common source of getting double-strand breaks.
Right. So previously we tried with some, but we tried specifically, I cannot remember, specifically probably the low doses of UV radiation probably. So and
UV would not give you a
but-
double-strand breaks.
yeah, but
Yeah, it will get.
No.
Either gamma would do,
So,
or
yeah,
any.
maybe probably we are using the zeocin treatment. So that
Yeah,
could
because
be-
after
yeah.
all, the plants should be exposed to the environmental thing, and one of the things could be the radiation coming down.
Yeah, there is
So
limited
that could
source,
also
yeah.
be.
Sorry, there is limited source of using gamma radiation, but instead the genome.
The
So that we are
method,
trying.
method of the break would be
Right.
different when you have an
Right.
ionizing radiation and the
Right,
chemical
right,
breaks.
yeah.
Okay.
Yeah,
Thank you. Let's thank Dr. Roy.
thank you.
Thank you, Dr. Roy. We come to the last speaker, Dr. Smruti Sanjita Behera. She is from IICB, Kolkata. She has been awarded fellowship from Ministero dell'Istruzione della University della Ricerca, Italy. And she's an international fellowship from CEDAT Empress International Graduate School. Her research area is dissecting the signaling components and molecular mechanisms involved with plant pathogen interaction in rice, investigating the role of calcium channels in pathogen recognition and immune responses in plants, understanding the function of sphingolipids in plant pathogen interaction, and studying the interaction between sphingolipid signaling and calcium signaling in plants during plant pathogen interaction. Dr. Behra.
Good afternoon, everyone, and thank you so for the nice and kind introduction. First of all, I would like to thank the organizers for giving me this opportunity to come over here and present the work I have very recently started at IICB. So, and also it's an honor to speak in a session which included my teachers and so many eminent scientists. So I joined IICB 2 years ago, roughly, and my lab became functional 1 year ago. So I have more questions than answers to show you, but it's also a good platform where I can get more suggestions and criticism from you so that I can be sure that I'm on the right path. As ma'am in the morning suggested or said that it's important to figure out the right question. So I'll see if I have put forward the right question or not. So yeah, basically I was doing, all my training has been in Arabidopsis, and I have been following calcium signals in abiotic stress conditions and understanding how calcium signal is perceived in Arabidopsis during abiotic stresses. But when I came to India and started thinking about a valid question, so it was important to include some organism or some plant that has either economical value or crop value. So I also moved to rice system and got interested in plant immunity and started to think about the interaction between rice and its pathogen. It's a gram-negative bacteria, Xanthomonas oryzae, Pathover oryzae, or in short, I would be calling it ZOO. So it's a gram-negative bacteria, and it's a hemibiotroph. It causes the bacterial blight of rice. This is, yeah. So basically, it is, as I mentioned, it is a hemibiotrophic pathogen, and it enters the plant cells through the natural openings or wounding, and it spreads through air and water, and it can spread very rapidly and has devastating effect on the crop production. So coming to the question, before coming to the question I would like to address, I just gave a brief introduction, which I don't need to explain here to this audience, I guess, but just for the students, a brief introduction, that basically we can divide the plant immune response into two categories, the pattern-triggered immunity or the effector-triggered immunity. So this is the first, when the first encounter happens with a pathogen, the plant tries to resist it by recognizing the pattern, some patterns from the pathogen. It can be the flagellum, or it can be the EF tau, or any part, or the chitin, which can be recognized by the plant, by the receptors present on the membrane. And then there are a cascade of events that go on, and ultimately it can provide resistance. But if this fails, the second round of immune response that is triggered by the plant is through the effector-mediated responses. I'm not going to this part now. Apart from these two, I was interested in understanding the first signaling events that take place between the rice plant or the host and its pathogen. So in general, these are the major questions what our lab is trying to understand. So as I said, I'm interested in the early signaling. The apoplast is the battlefield of several for plant and the pathogen. Especially with the zoo, there are several secretory molecules. I'm also interested in the plant extracellular vesicles, which is released into the apoplast, and how they are involved in immunity, but I'm not talking about that in this talk. And the OMVs, or the outer membrane vesicles, how they induce plant immunity, and what are the signals that are involved, like the calcium and the ROS signals. And in addition to these two, I'm also interested in understanding the role of membrane lipids, especially sphingolipids, in perception of the pathogen. So coming to the OMV, so the gram-negative bacteria, they release the outer membrane vesicles. So these are basically pinched off from the outer membrane, and they contain certain outer membrane protein, and also few inner membrane proteins are there, and they are loaded with several other proteins like the virulent factors and PAMPs, and then they are released into the apoplast. So, and if I briefly say what has been reported about the OMVs in case of plants is that they can be internalized, and all this information has been gathered just in 2 to 3 years. So it's very recent knowledge that we are having in case of plant and bacterial OMVs. So they can be internalized into the plant plasma membrane. They produce ROS. They are involved in remodeling the membrane, or the changing the lipid order of the membrane. There are reports that they do alkalinization, and also activation of immune response and PR genes. So coming to the two examples where OMV components have been identified, mostly they have membrane lipids and LPS, the lipopolysaccharides, outer membrane proteins, several virulence factors, and certain enzymes to degrade the cell wall of the plant. So the first thing what we wanted to do in our lab is to do isolation of these OMVs from the pathogen Xanthomonas, so which has not been reported in this context. So we could isolate the OMVs from BXO43. So this is the Indian isolate of Xanthomonas, which was isolated from actually West Bengal, and it was kindly provided by Dr. Hitendra Patel from CCMB, who is also working in this field. And we could isolate the zoo OMVs, and here what you see is a NTA movie. So this is a nanoparticle tracking analysis, where we wanted to see if we have isolated the OMVs properly and the range of their size. So we could see that roughly the BXO, or the zoo OMV, is in the range of 126 nanometer. And here I show you the NTA of E. coli OMV. So these are the moving particles, and this is more of a mixture of OMVs of different size, whereas BXO OMV or zoo OMV looked more uniform, and we also visualized them through AFM and confirmed that indeed we have the OMVs, as we did not have the marker for the OMV yet. So the next question was, do OMVs induce a calcium signal? Because there are reports that they induce an immune response and they also generate ROS. So as I mentioned that I'm very much interested in understanding the calcium signaling. So the immediate question was if these OMVs, they are inducing a calcium signal or not. And if they are getting incorporated into the plasma membrane of plants, what are they doing there? So a brief, very quick introduction to the calcium signaling. So any kind of stress or stimuli or any developmental cue can produce a specific calcium signal, which I have been following in case of abiotic stress. And these signals are very specific, very temporally and spatially defined. So we call them as signatures. So these signatures are very stimulus specific. So if you plot the change in calcium concentration over time, they may vary in their shape, and also the localization, if it can be in the nucleus, in the mitochondria, or in specific nanodomains or microdomains of the plasma membrane. So using genetically encoded calcium indicators, I could follow these signals, and just quick examples. Here, this is the rice root showing response to salt, and here is this is the Arabidopsis root, and this is the rice root showing response to external application of glutamate. And in ISCB, I have established a method to use a red genetically encoded calcium indicator, which is called rGecko. So this is a calcium probe, which has this M13 peptide and the calmodulin domain. When it binds to calcium, the fluorescence increases, and we calculate the normalized change in fluorescence to understand the change in calcium dynamics. So as we have established this in Arabidopsis in ISCB with a specialized chamber where we can fix the leaf and give a treatment and follow in a live condition what is happening with the calcium. So in the rGecko transgenic background. So here I show you what happens when you apply OMVs isolated from BXO. I hope you can understand that there is something spiking in the leaf cells, and these are the calcium signals that are generated when we apply the BXO isolated OMVs in Arabidopsis. And when we analyze different cells, so there are many cells, you define the ROIs and plot the graphs, and we could see that the signature somehow looked like this. And this has not been reported so far. And this leaf, BXO-induced calcium signature looks quite different than the FLAG22 induced calcium signal, which has been reported. So they have a distinct calcium signature in the leaf. And we also checked it in the roots, and we could see that in roots also, they are inducing calcium signal, and we tried to follow the propagation of the signal, and you can see that different ROIs were chosen. And if you follow the graphs, then you can see that this ROI here, this corresponds to the number 5. Here, in addition to the first peak, there is a second peak, which is originating here and then propagating to the upper part. So we are analyzing these dynamics to understand what is causing the first signal and why there is a propagation like this. So, and at the moment we are also standardizing this calcium signaling, calcium imaging setup in rice. So far we are in the midway of generating the transgenic rice lines, but we have been successful in developing the transient expression of the probe, the rGecko, in rice seedlings, which also shows a calcium signal, but that is in the process of standardization and optimization. So I'm not showing that here at the moment. Then, okay, the first thing was BXO induces a calcium signal. And last year actually, in 2022, there was a publication in Plant Cell which said that bacterial OMB internalization changes the lipid order. So what they say that OMBs, for the first time they showed that the OMBs are internalized into plant plasma membrane, and this, they correlate this internalization with the increase in lipid order in the plasma membrane after OMB treatment. And also, they say that incorporation of OMBs, so they devised a method to measure this change in membrane orderness or disorderness. So if the membrane is more towards or in more ordered, then the chances of there are more nanodomain formation. If it is more disorder, then there is no nanodomain formation. That is what they suggested. So we also followed this and checked that was done in Arabidopsis. We tried to check it in our system with rice and with Xanthomonas OMBs, and we chose two different OMBs isolated from E. coli and OMBs isolated from BXO. And this is the rice root, what you see here. So there is a protocol where you stain the OMBs and treat it in the root cells, and you can see over time the internalization. So what we could see that in the mock, there is no internalization of the dye, but in case of BXO or the pathogen or zoo treated OMB treatment, there is more internalization of the dye that corresponds to more internalization of the OMBs as compared to E. coli OMBs. E. coli OMBs also get into the membrane, but there is a significant difference between E. coli OMB and BXO OMB. And in addition to that, so there was also a difference between two rice varieties. One is Khitish and one is Siardhan. Khitish is a susceptible variety and Siardhan is a resistant variety. And what we see here is that with BXO OMB, there is more internalization with Khitish and less internalization with the resistant one. And we also followed the kinetics of the internalization. So then the next thing was to check the lipid order and disorderness after OMB treatment. So for this, we used a dye which measures the general potential of the membrane depending on the orderness or disorderness. And it calculates a ratio and gives a image like this which can be quantified to say if the membrane is more ordered or disordered. So as you can see here, if the GP value, the GP values here, actually I cannot see from there. So with BXO, the blue box, and E. coli, there is an increase in membrane orderness, but with the control you don't see that. And if you compare the BXO with the Siardhan, which is the resistant one, and the Khitish, there is also a difference, what you can see here. Okay, so the conclusion is that the lipid order of plant plasma membrane we can see the similar effect in rice with its pathogen, zoo, and OMBs of both zoo and E. coli, they significantly elevate the lipid order of the plasma membrane in rice. BXO and E. coli were elevated, E. coli OMBs elevate lipid order in a similar way in Khitish plant, but in case of the CR plant, with Siardhan, which is a resistant one, the BXO OMB elevates more or significantly higher lipid orderness than the than the E. coli OMB. So this is so far the work in this direction what we have done, and I can conclude that the BXO OMB or the zoo derived OMBs, they elicit a calcium signal both in root and in leaf, they are also, they get internalized in case of in rice, and they also change the lipid orderness in the membrane. So, so this was part of the work, and we are also isolating the plant-released extracellular vesicles and isolating them and studying their internalization and trying to figure out the components of the extracellular vesicles, and also what is exactly content in the OMVs of BXO or the zoo. And also we are trying to differentiate between the calcium signals that is induced by OMV, by the LPS, which is a component of the OMV, and by different elicitor which are present in the OMV itself. So that will give us more idea. And also a temporal study we are doing to see if there is a correlation in rice with this internalization and this lipid orderness. And yeah, rice calcium imaging is on the way, and also we are currently following the ROS signals, similarly with the, together with calcium signals. So that's all for now. I hope I'll have more things to tell next time. With that, I thank you all. And yeah, a special thank to the students who took all the trouble to join. They joined a new lab which had nothing at all. They spent 1 year in moving chairs and tables and setting up the lab. And thanks to them, I could start my work. And yeah, the work I showed here mostly was done by Ishani, who is a very smart and very daring person, I would say, who can take up any challenge, and I really appreciate that. And Sandeep and Remika, they have different projects, but they are also helping Ishani, and they're working in a quite well-coordinated manner, which is good for me. And a frequent visitor in my lab is my daughter. Thanks to her also for all the understanding she has that whenever I have to leave her home without paying attention to her. So yeah, and thanks to all the ISCB members, especially Arunda and our new director, CIF, and my collaborators. And yeah, also a special thank to Nivedita Di and Maitri ma'am for encouraging me for what I'm trying to do, and yeah, giving me support throughout. Thank you all for your attention.
This lecture is now open for discussion. Any question from the students? The lunch must not have sort of reduced your capacity to think.
Not the Xanthomonas OMB. So actually we also tried with the first we are trying to dissect one by one. So major component is the lipopolysaccharide. So we first tried with the commercially available E. coli LPS, and that did not trigger any calcium signal. And we are now isolating the LPS from Xanthomonas.
What is LPS full
The
form?
lipopolysaccharide.
No, why are you thinking that OMBs are having only lipopolysaccharide?
No, they don't have only lipopolysaccharide. We do not know because it has a lot more things. So to understand the signature, what I have seen is not matching with any known trigger so far. That's why I'm trying to understand if it is LPS or if it is individual effectors, or if it is the individual PAMPs that are present inside the
If
OMBs.
I compare it with the exosomes, they have a lot of microRNAs. Exosomes has a lot of microRNAs.
Yeah. So we have also isolated, that's what I told, that we have also isolated exosomes from rice, from apoplast, and also the total exosome. And that work is...
No. My point is, if I compare the OMBs like exosomes has a lot of microRNAs. The OMB also might contain a lot of microRNAs.
Yeah, we are in the process of not only understanding the microRNA, also the protein part, what is there.
Part,
Yeah,
yes.
that's I mean, I need little more time to do that.
Or Nindita, you have?
Yeah. I probably have missed it or not, I don't know. You have used zoo OMVs and treated Arabidopsis with it.
Yeah.
I didn't get the logic behind it because zoo is a pathogen of rice, right?
Yeah.
So why would you do it with Arabidopsis first?
So first of all, we wanted to see at all if the OMV is inducing anything or not, because OMV also contains the FLAG22 flagellin. It also contains the EF tau. It also contains certain other PAMs which have been shown to induce a calcium signal in Arabidopsis. So there are few reports, and as I said, that the rice work is we are optimizing it. We do see some signal, but it is to start with and to see if there is at all something or not. We try to do that, and to address this question, we are also trying to get the zoo, the XCC, the Xanthomonas campestris, which is the pathogen of Arabidopsis. So that would be interesting to see if there is a difference or not. Host specific OMV and non-host OMV, if they have difference or not. Yeah. So these are, I mean, there are a lot of things that are planned up to be done.
Actually, it's very interesting. The part which I really, very interesting, I found that OMB internalization of E. coli was much less compared to zoo.
Yeah.
So does it indicate an active process by plant for selectively taking pathogen one, or the pathogen in enforcing the
So
OMB in the
it
host?
is not known. This is what we are trying to figure out. So that's, and we also, we are in the process of procuring the XA21, the rice line, which has complete resistance to zoo. So Dr. R.N. Sundaran has this, the near isogenic lines. We are trying to procure that. That would also give, this comparison is required to see if with host, non-host, and different varieties of rice. Yeah.
Any literature available whether any kind of effectors are released to the OMV or not?
Yeah, so they have effectors. Even so, there I don't remember exactly now, but there are effectors which are present, which are released. So in case of Pseudomonas, I think I showed you in that.
It is okay, no problem.
Yeah,
We can
so
talk later.
there are effectors, but I do not know exactly all what are there in zoo OMV very precisely, and what is. It might happen that one component is involved in the translocation or the fusion process, because we do not know. One is responsible for this arrangement of the lipids so that the nanodomain can be formed, and once they are released, we do not know. So these are how they are released also is not known.
Thank you,
Yeah.
very nice.
I have just a small query. When you are talking about the disorderliness that comes up, are you also aware of how much fluidity changes are taking place?
No, so far we started, but that's a very important thing to measure. So just now we established this one, and the next thing would be to check.
Yeah, because that could also be reflected
Exactly.
through the type of disorder. You can get a smectic, nemetic kind of transition also.
Yeah.
You know, like the glass transition and other things that can be taking place. That's a more physical part of it.
Yeah.
Sure. Ranjita has a question.
Is this gram-negative bacteria a threat to rice only, or there are other plants?
It has an alternative host, which I showed in the first slide. So this is the plant, Lersia hexandra. But other strains or other strains of this pathogen can infect dicots. But this is the major isolate or major strain that infects the rice.
Okay. So it does not have wide host range, is that right?
Not really.
If there are no other questions, let's thank Dr. Behra.
Thank you. Can I just remove this one? Yeah,
You want to disconnect?
Yeah.
Oh, they will do it. Okay. Just it to disconnect. Okay. Thank you, Professor Thakur. May I now request Professor Arun Lahiri Mojumdar sir and Professor Ashok Ranjan Thakur sir to kindly felicitate our distinguished speakers, Dr. Ashish Kumar Nondi. Professor Ashish Kumar Nondi. Professor Jitendra Thakur. Professor Anindita Sheel. Professor Maloy Das. Professor Shujit Roy. Professor Shruti Shonjita Behra. Thank you. Thank you, sirs. So we came to the end of the first session. Before ending, may I kindly request the program coordinator and assistant director, Department of Microbiology and Department of Biotechnology, Dr. Rajat Pal, to say a few words.
Good afternoon, everybody. So today, this is almost we are at the end of our sessions. All the speakers, I am very thankful to all the speakers. Actually, this should be a vote of thanks, but as we are not ending today, we have our programs tomorrow also. For that reason, just I am saying that thanks to our Honorable Vice Chancellor and Chancellor for giving us this opportunity to organize this beautiful conference. Thanks to Professor Lahiri Mojumdar. His constant inspiration was there behind all of us for having this a good event. And all the speakers. Oh, I forgot to take Professor Thakur's name. I'm very sorry, sir. Because each and every time sir was calling me, Rajat, ki holo? Abstract boy ki hoche? Rajat, etar ki hoche, otar ki hoche? Everything was under his, he was just monitoring all of us' duties. And very nicely organized by our very young and very active convener, Dr. Sujit Singh, and both the co-conveners, Dr. Shori Roy and Dr. Siddhartha Datto, our very own and very beautiful, gorgeous madam, Dr. Banerjee, Nivedita Banerjee, our MCs for today. Two HODs, Dr. Shomshubra Thakur Chowdhury and Dr. Atrey Ghosh, and all other faculty members, Dr. Amit Sharkar, Dr. Shudipto Shaha, Dr. Rochishnu Datto, Dr. Ashish Jana. Everybody helped a lot. All my students, all the volunteers who helped us a lot for this event to organize, and all the participants. And a very, very big thanks to all the speakers who have come. In spite of their very busy schedule, they have come here and they are sharing their views, their experiences, their research work to this younger and very young students in a very young and very new university. I hope, with a very big hope, I am ending this speech, that we can organize another conference like that in the coming year, and all the speakers to whom I am meeting today, we are again meeting in the next conference. So with that, I am ending, and probably we again have some events. Dr. Thakur Chowdhury, I guess, is going to conduct that. So yes, that Dr. Atrey Ghosh will take care. So now enjoy the next sessions and the programs. Thanks.
Now we will have a special event by one of our biggest sponsor of this symposium, by Mark, and the event is on importance of high purity water for fundamental research. So may I now request our head of the Department of Biotechnology, Dr. Shomshubra Thakur Chowdhury, to kindly come and conduct the event.
Good afternoon to one and all present here. Thank you, Dr. Banerjee. So after this scientific session, we are now leading to a session which is more product oriented. So as Dr. Banerjee has already mentioned, Mark today will demonstrate one of the important product which is on the water purification. And we all know that what is the importance of Mark, their products, their contribution in the field of science, in the field of research. Their topic is importance of high purity water for fundamental research. And And today, from Mark, we have with us Ms. Samla Singh. Samla Singh is a valued member of the Mark Lab Water Solutions team, working in the field of marketing expert for the South Asia Pacific region. She has over than 12 years of experience in customer handling, and today she will demonstrate their product, and also a brief presentation will be there, which will you all enjoy, I think. With this brief introduction, I want to invite Shamla Singh on the stage.
Switch it on. Okay, am I audible to everyone? Okay. Thank you so much for giving me a slot. So basically whatever talk till now you have listened, it's all scientific. I am a commercial person. I belong to trade of sales and marketing. So we are happy that we are going to launch one of our product which we have launched on 25th January 2024, and it's specifically targeted for cost conscious customer, but that is a small part. Before that, we will have some conversation on water chemistry. So Merck is an organization, and we say that science is at heart of everything we do. Science is a legacy brand. It comes from 350 years of experience, and out of which 50 years, more than 50 years of innovation, which we are talking in terms of water purification technologies, which we are launching. We will see the journey which is there for our water purification system. So our vision is reimagining water purification to power the science of tomorrow. So irrespective of your needs, irrespective of whatever, I think, excuse me. There is a lot of noise. Can you ask them? So I would request people to concentrate a bit. I am not going to take a lot of your time. I'll try to finish up in 15-20 minutes. So coming to our vision, reimagining water purification to power the science of tomorrow. So irrespective of what field you are, whether it is micro, whether it is molecular, whether it is pharmaceutical, whatever field it is, there is a lot of usage of water, and today we will see like what kind of water is being used for what kind of application. Our mission is to bring the leading expertise and pioneering lab water solution to evolving need of every scientist. So starting from a startup to incubators, to a scientific lab, to the pharmaceutical industry, the biopharma, even the testing lab. So we, as Molecule, as Merck, we are everywhere. Water. Water is the most commonly used reagent in the lab. We call it as a reagent, and it is one of the most important reagent which we are using in our labs. If anything goes wrong with the water, the overall time what a researcher gives for that particular application, the end result, everything gets impacted. So that's why we are telling that water is commonly a very common use reagent, but again it is a very important reagent for any laboratory. What could be the potential effects of a water contamination? Can we have 2-3 voices from the audience? If you have came across during your research work, if water is wrong, what potential impacts you have seen in your daily day-to-day application? Because I believe everyone is doing the research here. Any 2 or 3 voices, whoever want to tell us. Yes, sir.
If the pH is changed, if it is not a highly purified and if the pH is very much acidic, then whenever we run the protein gel, that does not work.
Right. So it means we are getting erroneous results. It is one of the impact. How about we listen from the scholars? Anyone from that side? We don't have much time. If anyone want to volunteer. Come on, everyone sitting there is a researcher. You know what go wrong. Anyways, we'll move ahead. So there are erroneous results. What sir was telling, if your pH is acidic, like what can go wrong exactly? You need to troubleshoot, because you will not know that actually the culprit was water, why the application or the result has gone wrong. Then you do repeat analysis. Then you use repeat reagents or media which are expensive, means you are also putting up a cost on those results. Loss of samples, and then difficulties in data interpretation which generally come when we are running HPLC GC. The adjunct peaks or the ghost peaks comes up, and you will don't know that the water was basically wrong there. Means due to certain contamination, that particular peaks are coming up. We will now know that what kind of contaminants a water could possibly have. So we say it is H2O. I think everyone is very well aware, probably more than me, because I value the kind of scientists, the dignitaries that are sitting here. But coming to the contaminants, we have categorized it at like 5 contaminants. First one is organics, then ions, bacteria, particulates, and gases. So these contaminants are the basic culprits when we say what exactly is going wrong in our interpretations. Like why our bacterial growth is not proper. Probably the water which we have used for making the media, I don't know how much pure it was. You will never know it. The injector pumps which are getting blocked due to the deposition of particulates and the bacteria while running the HPLC, it is also one of the possibility which is coming from the contaminants. The basic water purification technologies which we have used through ages are distillation and deionization. So distillation even now we are using, because that is the basic technology what we are using for filtering the water, getting the ultra pure water. And next is deionization. But in coming slides we will see that we also have advanced technologies of water purification through which we are actually getting the accurate and precise kind of water which we have to use for our application. The next is going to be one video which is all about the kind of the different types of water which we are using. Can you check the voice? It's not coming.
Know your lab water. Did you know water used in labs is categorized as either type 1, type 2 or type 3? Why do different applications require different water qualities? Let's take a closer look. Water is categorized as type 1, 2 or 3 based on quality parameters which help to ensure we use the right water quality for specific laboratory applications. Type 3 water is used to rinse glassware, to fill heating baths and for other less critical applications. Type 2 water is used for general lab use, such as in buffers, pH solutions, culture media, and to prepare reagents for chemical analysis or synthesis. Type 1 water is the purest water and used for sensitive lab applications such as HPLC, ICP-MS, atomic absorption, and mammalian cell culture. For greater results, reproducibility, and reliability, labs should generally rely on types 1 and 2 water qualities for their analyses.
So basically there are 3 types of water, type 1, type 2 and type 3, but the majority which we use in our laboratory is type 1 and type 2. So type 1 we generally call it as ultra pure, which we are using for our molecular applications and the analytical applications. 2 is something we generally go ahead and use it for microbiology in autoclaves, environmental chambers, ELISA buffers and washers. So basically when you are going ahead and doing a research, so for the scholars and the students, so you have to basically see what kind of water you have to use for that application, and the right quality of water will actually ensure the accuracy, the reproducibility and maintaining the kind of result which you want for your particular application. So with this we are coming to first spot question which we have kept. What are the key water quality parameters that need to be considered for microbiology and molecular biology applications? We will take 2 answers and we have some goodies for these answers which will be couriered to that person. We have to collect their names and addresses. So whoever will go first come first serve kind of 2 answers. Just raise your hand, sir.
Resistivity megaohms. You have to do the 17 megaohms, right?
No, I am not talking in terms of sir value. It's just the parameters. The resistivity, yes, it is one of it. Correct. Sir, scientists are someone who already know. Yeah, I just want someone from the scholars because I want them to answer.
So mostly deionized water, right?
Sorry?
Deionized water?
No, sir. I am talking on quality parameters. Like resistivity is one which defines the quality of water. What other parameters it could be? So one sir has already answered. We are going for one more. Means your scientists are taking the goodies. Come on, guys. Yes, correct. pH is one. So pH, conductivity, resistivity and TOC. It is a very important parameter when we are actually looking for the purest form of water, going for trace elemental analysis. We go for water which is valued at less than 2 ppb, so that is too technical. So that is the kind of parameters we look into. So, sir, we need your and yours name. Okay, you can assign them. Yeah, correct. You have to nominate. Thank you so much, sir. Okay, so now we have seen the contaminants, we have seen the water types. Now we will see what kind of filtration technologies are in place which are used for purifying the water. So coming to the modern techniques, so the basic ones you have seen are distillation and deionization. So the modern techniques, we have the filtration, depth, screen and ultrafilter. So depth generally are used in the pre-treatments, which we are using also in our houses. So certain type of mesh is there of certain kind of size, and those colloidals and particulates are filtered. Screen filters are generally used in the polishers, are more refined, probably like 0.22 micron, and the ultra filter are basically one which we are using in removal of DNAs and the RNAs in our water. The activated carbon used for filtration of chlorine. In UV, we have two types of UV. One is bactericidal UV and one is photooxidation UV, which is used for purification of the water. We have RO, and this is something which is patented by Merck lab water solution. It is called an intelligent RO. So we know that more than 99-97% of the rejection happens at the water level. But here we also have a feedback mechanism where 40% of the water comes back to the source and again it is filtered. So we have certain set parameters. Let's say 40 microsiemens of water will only go through the RO and then to the next stages. Then we have EDI, electro deionization. Deionization is a process removal of the ions where we use cartridges. Electro deionization is somewhere we use the electrical field, no chemical regeneration happens, and year on year you get consistent water quality. And then we have IQ Nano, that is again a patent filtration technique of Merck. It is an ion exchange resin. Again it is used in our polishers to polish the type 2 water. So coming to the advanced water purification technologies, it's not one technology we are using for purification, it is combination of technologies always. So tap water to type 2. So type 2 is something which we are using for your microbiological applications. So from tap it goes to the pre-treatment pack, then to the EDI, the bactericidal lamp inactivate the bacteria. That water goes and gets stored in the tank, and from the tank you are taking the water 20 liters, 15 liters, 30 liters, 40 liters based on your application, and then comes the polishing stage. So type 2 water is being used as a feed and it gets polished through the photooxidation lamp where oxidation of organics happen, further purification of organics happen there, then the polishing cartridge, and then it goes to the final dispense point from where you take the ultra pure water. So you will see how exactly the purification is happening. So in pre-treatment, the chlorine means ions are getting, the organics and particulates are getting filtered. In RO, all organics, ions, particulates, bacteria are getting filtered. In EDI, only the ions are getting filtered. So likewise, it is likewise the filtration process is happening. One very important thing, one additional spot question for the students, means they are busy in their poster, but still, do we store our type 1 water? Do we store our type 1 water, or can we store our type 1 water? Looks like no one is ready, want the goodies, but still if anyone want to answer. Is it possible to store the type 1 water? No? Why? Okay, so basically we cannot store our ultra pure water. It is always freshly prepared. It is only type 2 water which we can store in the storage tank, which is advisable to be used within 2 to 5 days. Ultra pure water is very thirsty water. The moment it comes in the atmosphere, it starts attracting the carbons and the pH level, the TOC increases. That's why when sometimes we do the one thing which generally we use, that we test the pH of ultra pure water. So many a times researcher complains the pH is always come higher. Obviously it will come higher because there is a particular process through which you can actually test the ultra pure water's pH. So that's why it always comes from the point of dispense. It cannot be stored. So it is just one example we have shown. So DEPC is used for basically RNase free water, but the same quality of water you can have even through the ultrafiltration without using any DEPC, and you can have the similar bands through the ultrafiltration too. So whatever water purification we are using, it shows that it helps in removal of the RNAs and the DNAs, basically the nucleases from the water, the pyrogen free water, which you can use for your molecular application. There is another spot question. What is the minimum resistivity requirement of water for microbiology application in laboratory? Can anyone answer it? Should we skip the question? Okay, so the minimum resistivity which we use for microbiology application is 5 to 15 mega ohm. So that is the resistivity we use for the pure form of water, that is type 2. So this is a quick look of the specification of the type 1 and type 2 water. So whenever you want to procure or you have the system, you should check that the type 1 system or the ultra pure system should always give a resistivity of 18.2 mega ohm centimeter. If it's not giving that resistivity, that means your water is not ultra pure. And for type 2, it is less than 5, it is more than 5. Generally 5 to 15 is the industry standard we follow. So if it is like 10 to 15, it's fantastic, but even 5 to 15 works. And TOC for type 1 is less than 5, type 2 is less than 30. So when we talk on the TOC part, it doesn't make a lot of sense when we are doing the microbiology and molecular application. But the moment you go for trace elemental analysis, we talk a lot on the TOC part, because if your TOC is high, then the exact result of the chromatography could be affected. Coming to the selection of the water purification, definitely we have versatile solution. We are one of the pioneers in laboratory water solutions, coming from 1668 till now. So for again the tap to pure water and pure to ultra pure, and then also water for trace elemental analysis. We will have a quick look on the journey. So before like 1967, we are using distillation and deionization. Distillation is something we are still using, but in Merck, it started from 1967. We had Super Q, then Milli Q Ultra Pure, then we have started the chain of Ultra Pure, then compact. Many of the scientists could have used Century also, and then Milli Q Integral was one of the most finest design and the launch we had in Milli Q. Now we have 7th generation machine, more compact in size, the cartridges are more compact, the purification is fast, more advanced in terms of the data integrity, data connectivity. So Milli Q IQ 7000 we launched in 2017. In 2021 we launched Milli Q EQ 7000. In 2022 we launched EQ 70XX, that is a combination of type 1 and type 3 system. Now today here we want to launch a system which I told you that we basically designed for the cost conscious customers, specifically the incubators, the startups who would like to have a good water purification system for their applications. So this is Milli Q SQ2 series. It is very compact. We have kept it at the last so you can have a look and feel of that particular product. So it is that innovative and that ergonomic that you can actually take out the tank, and you can take out to the place where you want to actually do your basically you can take care of your application. This is a production unit, that is a basically polisher unit. So from where you get the ultra pure water. So basically it comes with an unlimited scalability, but this system is specifically designed where the water volume are less to be used in the laboratory. For higher volumes, definitely you have to go for the the upgraded models. It is ultra compact. You can just see it is just the size of your A4 sheet paper. You can install it yourself. You don't need an engineer. It can be done. It's just a plug and play kind of equipment. You just have to plug it. It is very simple, very ergonomic handset, very intuitive interface. So 18.2 you will see on the, it has a LED light where you could actually see it. Indeed a robust and reliable solution because it is coming from Milicu side. And then we have several patents behind this because this is a product which engaged 130 scientists, means it has engaged also our scientists through whom, our customers through whom we are doing the business. At the same time, the scientists, we are situated in France. So total 130 scientists from 2018 worked on it, and 2024 we have this launch. And it is sustainable. So we also have DFS scorecard designed for sustainability scorecard for this particular system. So it can be basically, you can actually even throw it or you can dispose it in the environment. Let's have a quick look on the video for this particular system. So thank you so much. That's all from my end. Thank you so much.
Thank you. Thanks a lot for the wonderful presentation. Now we will commence our poster and interactive session. May I request the head of the Department of Microbiology, Dr. Atrey Ghosh, to commence and conduct the poster and interactive session. Thank you.
We may proceed for the poster session that is behind this hall. And some of the posters, we could not find the owner of the poster, the presenter of the poster. That is poster number 21, 23 and 28. Please, the presenter, I request all of you to stand with the poster so that the judges comes and judge you. Okay?